In this video, we address the topic of expression. To study the function of genes, they are isolated from the original organism. They are then cloned into modified vectors and expressed in laboratory strains. The properties of the vectors in laboratory strains can specifically influence the expression of the target gene. Gene expression, or just expression, describes the process in which genetic information from DNA is transferred into a chain of amino acids. The conversion of DNA into mRNA is called transcription. The process in which the mRNA is used as a template to produce amino acid chains is called translation. Transcription is catalyzed by an RNA polymerase, while translation is mediated by ribosomes. An important aspect of gene expression is transcriptional regulation. As the name implies, it refers to the targeted regulation of expression at the transcription level. Various mechanisms can regulate expression. In nature, there are simple and more complex regulatory systems that occur in different organisms, cell types, or life stages to specifically produce proteins. Examples include the differentiation of stem cells into various cell types based on gene regulation or the regulation of metamorphosis in butterflies. A simple and widely used method in molecular biology for transcriptional gene regulation is the Lac operon. In nature, the Lac operon encodes genes involved in sugar metabolism. The Lac-Z gene produces the enzyme beta-galactosidase. Beta-galactosidase breaks down lactose into galactose and glucose, or catalyzes the isomerization of lactose into allolactose. The Lac-Y gene encodes beta-galactoside permease. Beta-galactoside permease allows lactose to enter the cell. Lac-A encodes the enzyme beta-galactoside transacetylase, which can acetylate galactosides. In addition to the genes coding for enzymes, the operon also includes a promoter region, P, and operators, O. The Lac-I gene, located upstream of the Lac operon, encodes the Lac repressor. The Lac repressor is a regulator that inhibits Lac operon expression by binding to the operator and blocking RNA polymerase. When lactose, allolactose, or analogs such as IPDG are present in the medium, they bind to the Lac repressor, causing it to change its conformation and release from the operator. This induces expression. Additionally, the catabolite activator protein, CAP, enhances expression when there is no glucose in the medium. A starvation signal, the cyclic AMP levels, causes CAP to bind to the DNA and positively enhances expression. Without CAP binding, the Lac operon is expressed only weakly, because the affinity of RNA polymerase without the activator is relatively low. The Lac operon is considered a leaky system, as RNA polymerase occasionally produces transcripts, even when the repressor is present. This leads to minimal operon expression. In nature, this allows for production of beta-galactosidase, which helps in removing the repressor. In molecular biology, this system is used to express target genes by replacing the Lac operon genes with one or more target genes, potentially in the form of using a vector system. The PET expression system is an example of vector systems that utilize the Lac operon. Here the gene of interest is cloned downstream of a T7 promoter region, to which Lac-I, the Lac repressor, binds. T7 RNA polymerase is required for expression in this system. Similar to the naturally occurring Lac operon, RNA polymerase binds less efficiently in the presence of Lac-I, partially blocking expression. The required T7 RNA polymerase originally stems from the T7 phage. To produce it in E. coli, the strain must also contain the gene for T7 RNA polymerase, often integrated into the genome as the DE3 lambda prophage. To further reduce leaky expression of the target gene, the T7 RNA polymerase gene, coded in the strain, is also regulated by LAG-I. Here, the E. coli RNA polymerase binds less efficiently to the promoter in the DE3 lambda prophage when LAG-I is present, further controlling the system. When IPDG or allolactose is added to the medium, this is called expression induction. The sugar binds to LAG-I both on the genome and the plasmid. This allows E. coli RNA polymerase to transcribe T7 RNA polymerase, which is then translated and expressed. T7 RNA polymerase then binds to T7 promoter region and initiates transcription of the target gene, which is subsequently translated. Thus, the inducer starts a chain reaction that leads to high target gene expression. However, since the LAG system is leaky, some target gene production can still occur before induction. As previously mentioned, expression systems like the PET vector system require specific strains, known as expression strains. Expression strains are strains that allow for the production of specific proteins in large quantities. One of the most commonly used strains with the PET vector system is E. coli BL21 Gold DE3. The strain originated from E. coli type B, which has a natural LON and OMT protease deficiency. In E. coli K12-derived strains, these proteases degrade misfolded and heterologously expressed proteins. This prevents protein accumulation. The deficiency was crucial for the development of type B strains as expression strains, as it allows for better production of heterologously encoded proteins. As such, E. coli BL21 also lacks the proteases and has additional characteristics. It has a reduced galactose metabolism and a deficiency in DNA methylation. The strain is suitable for vectors with E. coli promoters, such as the TAC promoter. Variants of E. coli BL21 DE3 carry the lambda DE3 prophage in their genome. This integration enables the production of T7 RNA polymerase, making the strain compatible with PET vector systems. Some strains, like E. coli BL21P lysis, additionally express T7 lysozyme. T7 lysozyme is a natural inhibitor of T7 RNA polymerase. This further tightens the expression system. The Rosetta strains additionally carry the P-rare plasmid. The P-rare plasmid encodes tRNAs rare in E. coli. This improves the expression of proteins that contain codons infrequently used by the bacteria. Finally, we will look at E. coli BL21 Gold DE3 in more detail. E. coli BL21 Gold DE3 possesses the previously described characteristics of BL21 DE3 strains. This includes the lambda DE3 prophage, limited methylation, and inactive LON and OMT proteases. Additionally, the endonuclease 1 and A is inactive, reducing DNA degradation, and the cells have increased transformation efficiency. During expression, soluble proteins should be formed, but protein aggregates, known as inclusion bodies, can also form. Inclusion bodies are insoluble protein aggregates that form due to misfolding during expression. Overexpression of genes can increase their occurrence. Inclusion bodies are often inactive, and only in rare cases do the aggregates retain functionality. The formation of inclusion bodies can be influenced by the choice of strain, temperature, or expression duration. In practice, this can be altered, for instance, by lowering the temperatures, or inducer concentrations, or by utilizing lower copy plasmids. As previously mentioned, lac operand-based expression systems are often leaky, leading to basal expression of the target protein without induction. This can be exploited for screening methods by detecting the activity of the basally expressed enzyme. The images show a filter screening of E. coli BL21 Gold DE3 cells with a PET vector encoding an esterase on the left and an empty vector to the right. Basal expression of the esterase catalyzes a color reaction. This allows the identification of clones carrying the target gene. More on the topic of screening methods can be found in the video Screening.